Very, very pleased and honored to have these presenters with us this evening. We have Dr. Juliana Rangel from Texas A&M. And also, I need to add doctor now in front of Garrett Slater. I apologize, so I will do that. Dr. Garrett Slater, who recently passed his dissertation, had been attending school at Purdue and will now be soon starting his postdoc in October in Baton Rouge. So we're really excited to have them with us for this July mini conference. And I'm Melanie Kirby. I'm the vice president, serving as the vice president of WAS. And a few of our board members are with, which I have a little slide about them. So welcome everyone to tonight's program. Here's a slide of just our executive board members. And so we're really glad to have these folks volunteering their time, and especially for numerous years, participating in WAS and helping to share educational opportunities for beekeepers, not only across the Western region, but wherever they may be logging in from. So thanks for logging in from wherever you're at. Our program is divided into two parts tonight. We'll have part one on queens with Dr. Rangel, and then part two with drones with Dr. Slater. And so we'll take a little 5-minute break between the two, but we'll go ahead and I should do stop share. We'll go ahead and launch in so that you all can get your slides going. So thanks everybody for joining us. Oh, I'm sorry, I need to do the bio. I was like, cool, he's going, done. So I'm very pleased to announce our first speaker, Dr. Juliana Rangel, and she was born in Colombia, South America. She obtained her Bachelor of Science in Ecology and Behavior and Evolution in 2014 from the University of California in San Diego. And in 2010, she obtained a PhD in Neurobiology and Behavior from Cornell University in Ithaca, working under Dr. Thomas Seeley. She was a National Science Foundation postdoctoral research fellow from 2010 to 2013, working with Dr. David Tarpey at North Carolina State University. And in January 2013, she became the assistant professor of apiculture in the Department of Entomology at Texas A&M in College Station. So you're coming up to your tenure there, your decade of running that lab. And I had the great pleasure of going to visit Juliana actually in May for her Queen course, and it was great. It was really wonderful and such a good program she put on. And her students are always doing really cool research, which we have a recent graduate of hers gracing our group tonight as well, Dr. Liz Walsh, who's now doing her postdoc in Canada. Or you finished? You finished, awesome.
She's now at the USDA in Baton Rouge.
I didn't know this. Okay, I need to get out from under the rock, but I've had a congrats, Liz. Awesome, awesome. Yeah, and so Juliana was promoted to associate professor with tenure in 2018, and her research program focuses on the biological and environmental factors that affect the reproductive quality of honeybee queens and drones, behavioral ecology and population genetics of unmanaged honeybees, and the quality and diversity of honeybee nutrition in a changing landscape. She's an active member of the Texas Beekeepers Association, and in fact, she got a really nice award of excellence from them most recently. And she's also participating in a lot of different associations. She used to also be, I remember, president of the American Association of Professional Apiculturists as well. And yeah, she teaches a variety of courses. I mean, Juliana has not rested, I don't think, since before I've met her. So her full bio is online, but we're just very happy to have her and grace us with some cool research information this evening. So thanks, Juliana. Go ahead and take it away.
So the first thing I want to say is that we are part of the at-home beekeeping webinar series, which is something that we also created during COVID. It was organized by Auburn University in particular the extension service at Auburn. Jeff Williams and Allison Shabel created this out of necessity for people who wanted to do bee talks and were stuck at home. So it was called Stay at Home Beekeeping Seminar Series. But it got so popular, like probably your mini conference, that we turned it into the at-home beekeeping webinar series, and we expanded our group to encompass not just SEC related universities, but also other USDA labs and universities in the Southeast, including North Carolina State, University of Georgia, University of Florida, Louisiana State University, and the usual from the beginning, the group at Auburn and Tennessee, and of course the Extension Service from Lawrence County. So it's every month at 6:30 p.m. Central Time on the last Tuesday of the month. So the last one was on June 28th by Dr. Paris from the USDA. And now the next one is going to be on July 26th. So that's next week by Dr. Rowe, dealing with Mother Nature, beekeeping in the extreme. So that should be very interesting because it's about changing weather patterns and landscape, etc. So that would be really great. And then we have planting and managing wildflowers for forage. And then the next talk will be by my most senior graduate student currently, Taylor Reams, who does work on the behavior and chemical ecology and genetics of Varroa destructor on September 27th. So you can watch it live on that link that you see there, Zoom link. You can watch it on Zoom, you can watch it on Facebook Live, or you can watch it after the fact for up to 2 weeks, pre-recorded on Facebook. So the fastest way for you to get to this is just Google at-home beekeeping, and it'll pop you to this page, and it'll either take you to the Zoom page or the Facebook page. So you scroll down, it'll tell you the schedules and how to link to the Zoom or Facebook pages. And again, they're totally free. And one of those convenient things about COVID was that now we can see talks from all over the country and the world from the comfort of our own home and for free. So I encourage you to join us. So um, when we talk about the factors that impact the reproductive health of honeybee queens, and you could argue the same for drones, you can divide these into two categories: biological and ecological or environmental factors. So some of the biological factors and biological traits that we measure in queens include mating frequency, which is the number of drones that a queen mates with. Insemination volume refers to the amount of seminal fluid, including semen, and sperm, but all the other components of the seminal fluid that the queen stores in the spermatheca upon mating. The age of the larva when initially chosen to be raised as a queen, and that we've shown in our previous work that if the workers or the beekeepers take longer to start the queen rearing process, and meaning how long they wait to start feeding royal jelly to diploid larvae, then the quality of that resulting queen decreases tremendously, up to 30 to 40% drop in productivity if the longer you choose to raise those larvae as queens. Other things that impact queen reproductive health include the pathogens that they either get from their drone mates or from through the fecal oral route from nurse bees that feed them. The microbiota that resides in their gut, especially the um, beneficial one that helps digest food properly. Her sexual competitiveness, which refers to how often she will go out and try to mate, how long she stays in those mating flights, and how successful she is at getting to mate with good quality drones. And upon mating, not just the volume of the sperm that they acquire, but also the count of sperm that resides within that seminal fluid, the viability of the sperm, meaning what percentage of the total amount of sperm is actually viable and could inseminate or fertilize, sorry, an egg, and motility and other things related to sperm quality. In terms of environmental factors, one of the most important ones is the quality of the nutrition that the queen and the workers receive in the colony. Exposure to pesticides that are used in bee pollinated crops, including, of course, agrochemicals, fungicides, insecticides, herbicides, changing weather patterns, and more now we're seeing big problems with extreme temperatures and their impact on queen and drone health, and the work that we will touch on today a little bit, the exposure to wax and food contaminated with hive chemicals during development, which of course was partly or in large part the work done by Liz Walsh when she was in my lab. So most people that talk about drones or queens have something like this graph where you have your queen or drone in the middle and you want to assess queen quality, and you have all of these factors that I just mentioned plus others, for example the age and the vigor of that queen and or her drone partners, the number of drones that are available for mating at the beginning of the season particularly and toward the end of the season, the genetics of the partners that the queen mates with, etc. But for the sake of this talk, we did in our lab some work related to our own, how our own beekeeping practices, which is the use of beekeeper applied chemicals to control the varroa mite on queen quality. But before we go further along, I want to talk about the role of QMP in the hive, because a lot of the work that we've done has to do with changes in the composition of QMP in queens. So QMP, or queen mandibular pheromone, is produced by the queen mandibular glands and spread all over her body via grooming. But it's not the only queen-produced chemical. In fact, the mandibular glands of the queen and other glands produce more than 3 dozen or 4 dozen chemicals. QMP is a blend of at least 5 chemicals. Some of them are semi-volatile carboxylic acids, like the 9-ODA, and also 9-HDA, the 2 enantiomers, and 2 aromatics, HOB and HVA. Those are the 5 main components of queen mandibular pheromone. And so it elicits mostly a retinue response, or a response of the worker bees that are taking care of the queen. They're called the retinue workers. And so imagine that the queen releases those all of those gland-produced chemicals, and they come out through, and she spreads them through self-grooming all over her body. Imagine these little dots being molecules of queen-produced pheromones, not just QMP, actually. I'll briefly mention this. And then all of these workers that take care of her are antenating her. Some of them are feeding her because they're the nurse bees that tend to the queen. They feed her, antenate her, and pick up all of these molecules of pheromones. So when the worker grooms herself, she will move most of these molecules of queen pheromones toward the front of her forelegs and their antennae. They will also start doing self-grooming, and so through that, there's a translocation of pheromones internally, and that's what causes all of these behavioral changes in the workers. But also through trophallaxis, they do reciprocal worker-worker contact that helps pass along the molecules of QMP, so that the queen doesn't have to come in contact directly with all the workers in the colonies. She just has to the workers have to come in contact with a certain titer, which we don't know what that is actually yet, but there's a certain minimum quantity of QMP that should be around for workers to either continue doing what they're doing or stop doing some of the things, and I'll mention some of those. Something really interesting, what I mentioned before, that it's not just QMP or queen gland-produced chemicals that maintains colony function, is the fact that the queens recently were found that queens produce also cuticular hydrocarbons. So they found some novel queen pheromones that are cuticular hydrocarbons and esters and acids produced by the charcoal glands. And all of these, either combined or separately, suppress worker ovary development. So that's one of the things that QMP is known for doing, is that it avoids, or it inhibits the development of worker ovaries and or ovarials, and therefore egg laying by workers. But in the absence of queens, you see these um, rogue workers that start laying eggs or unfertilized eggs. Well, in a recent study that was done by Tom Wenzelier's and his group, published in 2019, they show that they were able to reduce worker ovary development when workers were presented with synthetic blends of a full blend of QMP or just components of QMP, most importantly 9-ODA, 9-HDA, HOB, HVA. So um, alone or in combination, worker ovary development was inhibited, but interestingly, they also found the same type of inhibition with cuticular esters, cuticular alkenes, tergal gland esters, and tergal gland acids. So um, basically, the queen produces all of these chemicals that are not just produced by the mandibular glands, that are helping to maintain colony function. So as I said, the queen elicits QMP, and these queen produced chemicals um, elicit a retinue response and also inhibits worker ovary activation, but it also does other things. It inhibits queen cell production, it stimulates pollen foraging and brood rearing in small newly founded colonies. So this applies to first year colonies or first year swarms. And it also, in those swarms, increases nectar foraging activity and delays the age at onset of foraging, so that there is a larger population of nurses that take care of queen and brood at the beginning stages of these new colonies. And along the way, it reduces the production of juvenile hormone. It's still helping to maintain this youth in workers, so that they can help rear um, brood in newly established colonies. And in virgin queens, it attracts drones during mating. So we know that QMP composition varies among queens based on their reproductive state. So virgin queens smell very differently than mated queens, and in fact, that very special virgin queen odor is what helps drones locate virgin queens in the drone congregation areas. If you put mated queens up in the air, in those helium balloons, um, tethered to the helium balloons, you won't see the same attraction of drones toward mated queens, because they smell very differently. QMP composition also varies among queens based on development. As I mentioned earlier, the QMP composition of queens that were reared from 1-day-old larvae is very different in chemical composition than that of queens that were reared from 3rd-day-old larvae. And of course, we've also shown that queens that are reared in pesticide-laden wax also end up having QMP profiles that are very different to those that are reared in clean wax. So why do I care about queens so much? Well, the queen has this very specialized function in a colony. Her phenotype has been optimized for not only extreme longevity, but also extreme reproduction, an extreme form of reproduction where they lay up to 2,000 eggs a day, from only mating once or a couple of times with close to or a little bit over a dozen drone mates. And they receive the semen from their drone partners, and they store it in the spermatheca. Hopefully Garrett might talk about drone semen, and how it takes about 48 hours upon mating for all the semen that will end up going to the spermatheca to reach the spermatheca, and the queen then will never mate again, and she will lay eggs that are fertilized or not for the remainder of her egg laying career. And so it's very important um, to assess the factors that are helping these queens um, reach their potential. their highest fitness in terms of longevity and productivity. Now, changing gears, we know that based on the Bee Informed Partnership colony loss surveys, that colonies are being lost continuously year after year um, in the United States. There's an annual loss of approximately 30 to 40% depending on the year. And if you ask the beekeepers in those surveys, they always, in the last at least 5 to 7 years, have placed these five or four categories: poor wintering conditions, which means weak in the winter, which means basically sickened colonies for several reasons, varroa mite issues, pesticide exposure, and poor queen quality as one of the top reasons for colony loss. So I'm interested in that intersect between what we do to control the varroa mite using pesticides and how that affects queens and also drones. But for this talk, we'll only talk about queens. So we want to help our bees by using products that control for the varroa mite and reduce mite levels to below the 3% threshold of mites in the dispersal phase. But we know now that the mites have um, developed resistance to the most common miticides, including recently resistance to amitraz, which is the most commonly used harder chemical to control mites. And Frank Reykjavik in 2020 published a paper noting that there are pockets of mite populations in the US that are now resistant to amitraz. Um, and he actually, I just read in the bee culture newsletter that he is enlisting, I think it's 200 beekeepers from across the US to provide samples for him to test whether those mites across the country might be resistant to amitraz, which is troubling because that's the most widely used product nowadays. So we know that honeybee colonies accumulate contaminants that are detrimental to the health and productivity of colonies, and more we see work showing the interaction between pesticides, mite stress, and diseases that contribute to colony declines. In particular, again, I'm interested in those that cause issues in queens and drones, and we know that tau-fluvalinate, the pyrethroid, um, that's the active ingredient in Apistan, and then Coumaphos, the organophosphate in the product, Checkmate Plus, were shown separately to cause issues with queen reproductive health back in the 2000s. Most of those studies, though, were done testing one or the other independently or at very high concentrations that were applied topically on top of the mature bees' bodies. And even then, they showed that these queens ended up having low ovarial weight, low sperm viability, higher mortality. Um, but we are more interested in seeing how these chemicals affect queens when they are still developing, because they might come in contact, the larvae may come in contact with contaminated wax while they're being fed by the nurses. And so I argue in my whole idea about this type of work was that the wax is contaminated with chemicals, and then the nurse bees will feed the developing larvae, and the brood food is liquid, and some of these chemicals will leach into the brood food, and therefore the developing bee is going to consume contaminated food. And then that's going to have downstream implications for adult quality and mortality. So we know that even today, even though this study was done a long time ago now in 2010, we continue to see fluvalinate, coumaphos, and amitraz as the most commonly found miticides in wax. We also found uh, a lot of insecticides and troublingly fungicides in wax matrix at levels that should be of concern to beekeepers, especially because we don't have the same regulations for bee health in terms of approving fungicides and herbicides as we do insecticides, and yet we now know that chlorothalonil, for example, it is causing a lot of health issues to honeybees. And so we found um, these chemicals still to this day in newer studies published in 2016 and then in 2019, they persist in the wax matrix even to this day, even though we don't use those products anymore. We only use amitraz mostly. Although I've heard some people started using Apistan and Checkmate Plus again because we stopped using them. So now they are again effective in some mite populations. So I'll talk about basically the thick of Liz's dissertation of what aspects of honeybee queen physiology and behavior are affected by being reared in wax contaminated with agrochemicals. And so we have several aspects of queen biology that we looked at: morphology, ovarial size, chemical composition of mandibular glands, retinue size, egg laying rate, sperm viability, mating frequency, and most recently, um, gene expression in the brain of these queens. And so for all of these studies, we kind of followed the same procedure, which is to rear queens artificially, doing the grafting procedure, which helps produce queens en masse for most queen rearing operations across the US. And so you end up creating these either plastic or wax cups into which you transfer a 1-day-old worker larva, and then you put them into what is known as um, cell builder, which is a queenless unit of bees of nurse HBs that helps raise queens when needed. And so for these cups, we coated them with the approximate amount of wax that you would have on a regular wax cup, which is about 200 milligrams. And we had this molten wax covering the interior part and basically the whole coating of the cup. Um, and we either had them as being pesticide free, or we had spiked the molten wax separately with combinations of these chemicals that I showed from that table. The two previously used miticides, fluvalinate and coumaphos, the currently used miticide, amitraz on its own, and then the top two agrochemicals that were found in that study together at field relevant concentrations. And then we allowed those queens. Um, so we grafted again 1-day-old worker larvae. We introduced them into those queen cells to queenless cell builders. That's Liz grafting, so moving the 1-day-old larvae from the worker brood nest area into those plastic cups. And then we used queens at different stages of development for each variable measured. So for most of the variables, we used them as mated queens. So we allowed the queens to, once each of these queen cells was sealed, we placed it into an individual mating nuke, which is a unit of bees that you put into a small box, and you feed them and let them habituate to the queen cell. And once that queen emerges, then she will take her time to roam around the colony, then do her orientation flights, and then do um, her mating flights and come back. And you want to ascertain that she has successfully mated with drones by waiting until the eggs that she lays are capped, and that you see that the cappings are flat and looking just like regular worker brood before you use those queens for any other measurement. And then for a very small subset of queens, we created some that were sacrificed as virgins for morphometric analysis. So we did morphometrics on virgin and newly mated queens once we had learned that they were indeed successfully mated, and measured the typical things that people do when they want to keep queens alive, but they kind of want to see how good they are. So they measure body weight, head width, and thoracic width. Some people go, above and beyond. They may measure the length of certain either leg parts or wing parts, which all correspond to basically size. So for all of these graphs that I'm going to show you, we're going to have the treatment group on the x-axis, and so we're going to have control, meaning the queens that were reared in the pesticide-free environment, and then we have the other treatment groups. Fluvalinated coumaphos is when we added those two together to the wax. Amitraz, we added amitraz only to the wax, and then chlorothalonil and chlorpyrifos is what we added to the wax on the last uh, agrochemical group. And then on the y-axis, you're going to see the trait that we measured. So in this case, for example, body weight of virgin queens. Oh, and then inside each of these bars, you're going to see numbers or n equals something. That means the number of queens that were measured for each of these traits. Some are going to be higher or lower based on the availability of queens for each particular treatment group. So no significant differences in virgin queen weight. There was a trend of lower weight for the amatraz queens compared to a control, but it wasn't statistically significant. And no differences in mature queens. They were recently made and maybe within a month after they had emerged. Obviously, you can see that the average weight of the virgin queens was about 75 milligrams lower than that of mature queens because they develop their ovaries and they become bigger when they're mature, but no differences in mature queen weight. We also looked at worker retinue size and queen egg laying rate. So once those queens were analyzed for morphometrics, they were placed in a 3-frame observation colony like the one you see here. They were placed at random into observation hives, and they were caged so that they got accepted by the workers. And about 2 to 3 days later, we uncaged them. We allowed them to roam naturally, and around the next day, when we knew that they were fully accepted, we took the data. And of course, you know that I'm talking about the institutional we, which is that Liz did all the work um, and her assistants. But anyway, that's how we go. So we measured. So we introduced these queens into observation colonies that had about roughly 5,000 to 7,000 workers. You can calculate that a fully covered frame or a frame fully covered with workers on both sides has roughly 2,000 workers. So that's how you calculate or guesstimate the population of an observation hive. We measured worker retinue size by counting the number of workers per minute that were found around the queen for 5 consecutive minutes. So you can see on the bottom left here a very robust queen retinue with a lot of bees being interested in the queen, and that's a very good indication that that's a very good, attractive queen to the workers. And she is um, getting fed more often, and she is better liked by the workers compared to a queen like this one here, where there's very little attraction by the workers, a retinue of roughly 3 bees and nothing to go home and talk about. She is not being fed as often or as regularly or as much, and it's probably because her smell, amongst other things, probably behavior too, but her smell is it smells weird to them. We did point sampling, which means that you look for the queen and you do these counting observations for a minimum of 55-minute bouts per queen. And we also measured queen egg laying rate, so that counted the total number of eggs that were laid per minute for a 5-minute stretch by each one of these queens, and we did a minimum of 55-minute observations for queen egg laying rate. So in terms of the retinue, we found that there was a significantly smaller number of bees um, taking care of the queen when she was reared in any of the 3 control groups compared to the control group. So um, the average was almost 15 queens in the control um, group. So imagine there will be 15 workers around the queen, just like it's shown in the bottom left graph. Um, for the control queens, whereas there was a significant drop in the number of workers that were interested in the queens that were reared in either amitraz, C plus C, or fluvalinated coumaphos. So that has big implications for queen feeding behavior and the workers' attractiveness to the queens. In terms of egg laying behavior, look at this very troubling picture. You have on the y-axis the number of eggs that were laid in a 5-minute interval. And of course, you have the treatment groups on the x-axis. And so you have roughly 2 eggs per 5-minute interval in the control queens, but that number drops dramatically for the F plus C group and the Amatraz group. There was no difference between the control and the C plus C group in terms of egg laying. What does this mean though? Because who lays 1.3 eggs? That's biologically doesn't make much sense, but it's because of the type of data we were taking. If you extrapolate those data and you assumed that the queen is laying roughly the same rate at roughly the same rate for the entire year, which we know is not true, but let's say at maximum by extrapolation, these queens that were reared in the control group would lay roughly 200,000 eggs a year, whereas queens that were reared in this group would lay only about 360 eggs a day or 132,000 eggs, which is a decrease of 35% compared to the control queens. And in the Amatraz group, they would only lay about 115,000 eggs a year or a decrease by 43%. So just grafting queens in Amatraz laid in wax can drop a queen's egg laying capacity by 40%, and I think that's really, really dramatic. We also prepared the queen's mandibular glands. So after all of these observations with live queens, we sacrificed the queens. We took them to the laboratory, very precious queens. And so we wanted to do a lot of things with these dead queens. And the first thing was preparing their mandibular glands for extraction of the contents of the gland. So this little teardrop looking thing here is the gland, and the contents are kind of almost like it's like an oil, very beautiful shiny yellowish oil, and you put the contents here and you do chemical analysis. So what you would do is extract both glands and used one gland for chemical analysis and the other gland for behavioral bioassay that I will talk about shortly. And so for the behavioral bioassay, you have to collect 1 day of workers, emerging workers. So you take a frame of emerging brood from a colony, you know that they're emerging, so you brush off any other bees, you put that frame with food in an incubator so that 24 hours later you come back and all the bees that are on that frame, you know that were born within the last 24 hours, so you can age the bees. And we know that nurses start performing these nursing behaviors at around days 5 to, within that week of 5 to 10 to or so after emergence. So 5 days later, we would take these cohorts of nurse HBs and we put them in these cages that would contain roughly, I believe, 30 workers or so, and we would take the contents of these, one of the glands per queen, either a control queen or an experimental queen, and we would smear the equivalent of half of a queen's mandibular gland on each one of the slides. So you would have a control queen smear on one slide and then an experimental queen's smear on another glass slide, and would count the number of bees that got interested in the contents of those glands, because we know from previous studies that nurse bees are really attracted to the queen's glands contents. And then, as I said earlier, we also did the chemical analysis of the queen gland contents, the compounds using GCMS, gas chromatography mass spectrometry. And Liz learned this whole technique with collaborators at Texas A&M to do all the chemistry by herself. And so she took, she had the glands from 17 control queens, 8 F plus C queens, 15 Amatraz queens, and 10 C plus C queens, and did the chemical analysis for each one of those. And as you can see, That's why I said at the beginning that the queen mandibular gland contains more than like 3 dozen different chemicals, some of which are unknown. So they not only include the known QMP compounds like 9-ODA or HVA or HOB, but a group of other chemicals that we still don't know the function of very well. But we basically, to kind of summarize the all of this raw data, we compared the relative amount or concentration of each one of these compounds per queen, and you immediately see that there's differences in the relative concentration of control queens compared to um, uh, F plus C queens compared to Ametras queens. And so if you take all of those, each one of those components together separately and put them into, instead of an X by Y plot, you have an X times Y times Z, several different layers of analysis. You do what is known as a principal components analysis, which allows you to put all of those different compounds into one sole analysis that looks at the differences in chemical composition across queen types. And we found that indeed there were significant differences across the board in the chemical composition of the gland contents. And the first component of this first statistical principal component comprised 5 identified compounds that amounted for 80% of all the variation that was observed across treatment groups. So basically, long story short, control queens smell very differently than any of the experimental queens, and that is seen in something like this in the bioassay, where you look at the number of bees that are attracted to each slide. If you take the difference between number of bees that are attracted to control queens versus the smear of the F plus C group, you see a drop of almost 40%. So a significantly lower number of bees is attracted to the number of to the contents of the glands of the F plus C group compared to the control. You also see a drop of about 20% in the attraction of workers toward the gland contents in the Ametras queens compared to the control. Again, for the C plus C group, we did not see differences in retinue size. In attraction of nurse HBs between control and C plus C reared queens. We also looked at the number of ovarials per queen. So we took a cross section so we dissected the ovaries of each queen, the right and the left ovary. Then we took a cross section of the ovaries. We fixed them and stained them at the histology laboratory at A&M, and took photos and then counted the number of ovarials. So each one of these is what you would consider one of these ovarials. We first did the analysis doing both the right and the left ovary when we realized that there were no significant differences in the number of ovarials between the one queen's right and left ovary. We only used the right ovary for the remainder of the analysis to cut time in half. But we did not find any differences in the number of ovarials per ovary across queen types. So no differences. The number of ovarials ranged between 100 and 200 ovarials per queen. We also looked at sperm viability. So we dissected out the queen spermatheca like such. So we took out the tracheal net that covers the spermatheca, and we popped the contents in a diluent that, amongst other things, contained 2 dyes, cyber-14 and propylium which dye either live sperm cells green or dead sperm cells red. And then it allows you to get, if you add these two together, you get the total sperm count in a volume. And if you only take the green portion, then it gives you viability. What proportion of the total it actually is viable or fluoresced green. And we found no significant difference, a statistical trend in lower viability in the F plus C queens but nothing statistically significant because it has to be at or below 0.05 across queen types or treatment types. Finally, we did something to assess um, queen mating frequency, which is estimating the number of mates that the queen mated with. And this is done genetically, of course. You have to extract DNA from worker progeny from that queen because we don't have the queen drone partners in the colony to assess who she mated with. But through genetic tests, you can assess paternity because the workers are diploid, and so half of the genes come from the mom, the other half comes from the dads. And if you use these different um, software programs like Genius and Colony, you're able to then categorize each of these workers into one bin, and you get the whatever, however many number of bins you get per colony is the number of estimated paternity frequency or number of mates done by the queen. And what we found that was really interesting was that queens that were reared in the F plus C um, group had a significantly higher mating frequency than queens in the control group. Actually, queens in the amateurs group also had significantly higher mating frequency compared to the ones in the control group. Again, there was no difference with the C plus C group. So this was very curious, higher mating frequency based on the rearing environment. And it would look kind of weird to you, except that I had done a study in 2015 when I was working with David Tarpey's lab where I got exactly the same result. Queens reared in an F plus C laden wax had a significantly higher mating frequency compared to queens in the control group. And so higher is not always better. This is kind of out of sync with the queen's physiology. So something weird is happening in the queens. What could it be? So the last project, really quickly, is being finished by my current graduate student, Myra Dickey, who is looking at how gene expression in the brain is affected by being reared in wax that's contaminated with agrochemicals. So we took the brains of these very precious queens that Liz had taken, that we had all the behavioral data for, that we had all the genetic mating frequency data for, and we now took the brains and to wanting to characterize any kind of genes in the brain that are differentially expressed in response to pesticide exposure during development, and compared them between treatment groups to see if we can identify new novel pathways or targets for each pesticide that will affect queen reproductive output. And so when the queens were laying, again, we collected the queens. That's basically the same queens that Liz reared. And so we knew that they had been laying successfully. And then we dissected the brain out and isolated RNA and sent it off for sequencing. And so this is kind of, I don't have a lot of time to go over this. I just want to show what is called a volcano plot, which identifies differentially expressed genes, basically meaning which genes are more expressed or less expressed than random. So it has to be there has to be a statistical figure there. They have to be statistically significantly either higher or lower. So if they're higher, we call those upregulated compared to the control group. If they're lower, we call them downregulated compared to the control group. And so we have three different comparisons. In this case, the genes that were differentially expressed between the control queen brains and those in the C plus C group. And so anything that is to the right or the left of these and above these dotted lines will show up as a different color, in this case red, compared to the ones that are in black, which were not significantly different. And those are, each dot is a gene. And so here's the gene bank number for that gene. We found that for the C plus C group, most of the genes were upregulated. There were a total of 238 genes that were upregulated due to the queens being reared in C plus C wax. In F plus C, the same thing happened. We saw 241 upregulated genes caused by F plus C compared to control wax. And look at this very interesting flip of a coin for Amitraz, which is the one that we currently use and the one that I'm most interested in. Most of the genes that were differentially expressed were downregulated by Amitraz, and there were 618 genes downregulated by Amitraz, and 49 of those were upregulated. So uh, 3 times more genes were differentially expressed by Amitraz, and most of them were downregulated. So what we're doing now, finishing up, is we identified 5 genes that have to do with reproduction, because remember, queens that developed in contaminated wax laid fewer eggs in Liz's experiment. So is there something going on that there's miscommunication of between the brain and the physiology that is causing these queens to lay fewer eggs, potentially mate with more males than normal? And so we are looking at these uh, target genes that were downregulated by Amitraz, and see what their actual function might be with these queens. So in summary, we're finishing up the transcriptome paper. Should be done actually soon, within the next month. We're going to submit it. It's going to have the list of all the genes that are downregulated by Amitraz and upregulated by the other groups. We found some of these traits had were affected by agrochemical contamination, but not all. For instance, we didn't see changes in morphology or ovarial size or sperm viability, at least in newly mated queens. I think if we had waited 6 months, we would have seen differences in sperm viability. But for the rest of the traits, we did see that these chemicals um, are affecting our queens' physiology and behavior. So, of course, Garrett is going to talk about why we have so many dot drones, and I think and we have shown that pesticide contamination is one of the reasons why these drones are not as good as they should be. Um, Nutrition, I think, is one of the most important things that are affecting drone and queen quality. Changing weather patterns as well. So that's the kind of line of work that I am interested in continuing in terms of reproduction. So with that, I want to thank you for your time. Want to thank Liz for being in the audience, because I talked mostly about her work, all of our collaborators. I encourage you to visit our Bee Lab website. We have now almost 5,200 followers from all over the world, and counting. So if you want to learn about what we do in the lab, you should just follow us on Facebook, and we actually have an Instagram page that is the same, Tamu Honeybee Lab. So I'll take any questions after Garrett's talk, or via email at your convenience. Thank you very much.
Thanks, Juliana. And I just have to say, I love this photo because this is when Juliana came to see me in New Mexico back at, I want to say that was almost 10 years ago too.
I love that photo.
Nice.
Yeah, I love it.
So I just, there's just a couple questions in the chat, and I know one of them you touched upon a little bit about age of the queen, because you were saying that if you had waited, for them to be 3 or 6 months, might have seen a difference in the sperm viability. Do you want to touch upon that just briefly? Their question is, were the age of the queens considered when testing? Did results vary according to the age of the queen, and does
Yes,
pheromones vary
so
with age?
absolutely. There have been a few studies that have shown, and actually I intend to replicate that study here in the US. But there's one where they took a cohort of queens. It has to be a large cohort, and you have to have a large apiary to maintain, let's say, 50 or 60 queens, so that you have at least 20 in each age cohort, so that you have statistical power to test this. They took queens, and then they allowed them to mate, and they sacrificed a third, so they called those year zero queens, to look for sperm viability. Then they allowed the rest of the queens to make it through the next year. Whoever made it to the next year, they took half of those queens and sacrificed those for sperm viability, and then they allowed the other ones to go through for another year until they overwintered and a year had passed, and they called those year two queens. But and they showed a decrease in sperm viability of about 40 to 50% year after year in the queens. And so that shows immediately the problem of or why we see supersedure happening, because the queens are running out of sperm really quickly. Um, and and not to mention, I don't, that study was done in the Middle East, and potentially they don't use as many agrochemicals as we do, so the fate of our queens would probably be lower. They also showed that the sperm was slower in motility as years pass. So as we all senesce, oxidative stress causes things to slow down and to age. And so the age of the queen absolutely has uh, not everything, but a lot to do with her quality.
Yeah, thank you. Well, and that'll be cool when you replicate that here stateside to see what what comes out of it. Just sort of as an update too. Um, well, thank you, Juliana. And I know maybe some of our panelists might have questions for you, but I want to just go ahead and put real quick up for those of you who want to take a quick little bio break, you can, and then we'll come back with Dr. Garrett Slater too. But Juliana, the other question actually in the Q&A is about drones and about drone saturation. So I thought we might save that for Garrett, or if you want to take a look at it and maybe write something back about it, you can. But yeah, so for anybody who wants to take a little 5-minute bio break, you can do so. And so we'll kind of log back on here. I mean, we're all on. I'm not going to log off, but we'll go ahead, Randy.
Yeah, Juliana, it says interesting that Jim Bach from, I guess, Washington State years ago noticed is that fewer, smaller retinues or fewer retinues around queens. And we've had observation hives for, geez, well over 30 years, maybe 40 years at our county fair, which I've stocked that long. And back in the day, it was very easy to point out the queen to the visitors because they always had retinues around them. That's no longer the case. Now, here's the key thing. I haven't used a synthetic miticide in over 20 years. Our combs are very, very clean. So this is something that's happened independent, and we're in a non-agricultural area. So this is something that's happened independent of miticides or agricultural chemicals, at least in my operation. The other thing I'm seeing is we also have more, I don't call it queen failure, I call it failure to supersede. That back in the day, we didn't see much queen failure at all. And I wasn't keeping data at that time, but I remember I used to take loads of 72 colonies up to Nevada the 1st of July and leave them up there on the alfalfa until fall. When we'd go up to pick them up, we'd count on 71 colonies being queen right year after year after year. We don't see that anymore. We see a higher rate of queens failing. So, and I'm not saying those queens died. I'm saying that the queens are always being replaced and they're being naturally superseded. Colonies can very easily supersede their queens. That's a natural behavior of colonies. What I see more is what appears to be failure to successfully supersede. So that's my big question. And like we sell 1,000 nukes every year. And in nukes, we see in our own that we see, queens start off, they lay okay for a few weeks, then, some get superseded, but they successfully supersede. Superseder is a normal thing, but I hear more and more about this maybe, and again, I like to use the term failure to supersede rather than queen losses. And this appears to be independent of miticides or pesticides, but seems to be related possibly to varroa or viruses, because this has happened since them. You're muted.
Can't hear you, Juliana.
Yeah, that's a good point about viruses. When they started tracking queen events, they called it queen events, in the BIP surveys, they always noticed that the colonies that had queen events had higher viral loads.
Mhm.
And so it's probably
That's my number one suspicion.
yeah, pesticides are exacerbating the problem, but they're
Right.
not necessarily causing the problem. They're not the direct cause. It's a concomitant kind of situation of all of the above, all of the factors. And I
Yeah.
wish we had
I'd be
a way
interested in
to.
seeing studies from, say, the Isle of Man or Australia of virus prevalence. Oh, they probably have them from Australia. Liz, you know, have you seen any studies from Australia of viral prevalence in
Yes, they
in
have some, but
Varroa
they're
free
not
bees as
as
opposed
high.
to
Yes, right. Their viral loads are not as high. Do
right?
you, by the way, do you call this failure to supersede? So if you read Dewey's book, um, they have something that they call efficient supersedure and inefficient supersedure.
Hmm.
And intuitively for me, efficient supersedure would be that the queen got successfully replaced with another. But that's
Hmm.
not what they mean. Efficient supersedure is when you have a two queen colony.
Hmm.
Oh, okay.
So and you can have it for a while, two queen
Yeah,
colony. And
I've actually
so
seen grandmother, mother, and daughter, three generations in colony before one time, but two queens are because when we're grafting, it's not uncommon to find two queens
so
in
when
a
you're
colony.
saying
Yeah.
failure to supersede, you're talking about a two queen colony.
Not necessarily, but that you wind up with a queen does not give the signal, or the workers do not detect the signal to replace the queen, while the queen
Oh,
can
so
still
okay,
lay viable eggs.
okay,
If the queen is, I
yeah.
mean, if you remove a queen from a colony
Mhm.
can generally rear a new queen. But if the
Hmm.
queen as long as there's young enough larvae, diploid larvae, if the workers don't get that signal until the queen fails to having young larvae, that's a failure to supersede. The colony
Hmm.
goes queenless or laying
Oh,
worker.
I see what you mean. Okay, yeah. Still,
Yeah, there's oh, I didn't mean to interrupt you, Julie. I feel like there's so much more to
no,
talk
so
about.
much to talk about.
Mhm.
Yeah.
Yeah, and it makes me think, you mentioned Jim Bach, who served as the WASH treasurer for many, many years. So he was really active in this group. But he had also brought up frequency. He was really interested in frequency within the retinue in terms of what, he said that some of his measurements, and it would just be great for somebody to follow up on this, that those with, of course, low retinue had lower frequency. And then, of course, they didn't pull through winter. So we'll go ahead and let Medhat mention something, and then we'll switch over to Garrett. So go ahead. Thanks so much for the conversation.
Yeah, we have a lots of these issues also here in Canada, but recently just running a project, just looking at queens. Most of our queens comes from different locations around the world, and some local queens. I'm finding 1 out of every 5 queens imported, it has less than 700 to 900,000 sperm. How you explain that? Then I went out, and after we introduced queens for 6 weeks, I retrieved the queens back, sisters, and I looked at them. 2 out of 10 queens almost running out of sperm. So that really opened my eyes. Are we looking at the wrong question here? So our guys here who are producing local queens, keep in mind those guys like each guy, like I have one guy was putting 200 nukes in one location. His average sperm per queen, it was about 2.3. This year I told him just to put 20 nukes in each bee yard. Just I have the data here right now, 5.5 million sperm. Simple questions like
Yeah.
this one require simple answer, but we are always looking for exactly the same thing. Like, we're using a before. Like I get this question actually this year. we heard some of these talks, and Medhat, what should we use? Varroa mites or quality queens?
Mm-hmm.
So I said for raw mites, quality queens, like what you said, Juliana, that is, it's really great to know high sperm, high quality queens, you might really be able to overcome the issues in the hives.
Yeah.
But also, don't use illegal chemicals and contaminate every single thing. Our guys using the legal stuff, and I kind of, in a way, I would like to take some queen cups from the hives, treated here in Canada, and compare it to other places and see how much contamination in the queen cups, because usually it is made out of new produced wax. So how much of that chance of that queen cup will be highly contaminated is going to be kind of, I don't have data for that, but that's a good question to ask, actually. Just look at the queen cups in nature and queen cups coming from cells or colonies treated. So we can get a little bit more information about what's happening out there. But really, right now, everything is starting to go out of my head. 1 out of 5 or
That's
2 out
crazy.
of 10.
That's crazy.
Yeah. Yeah.
Yeah, there's so much that we can learn and study. So thanks so much, Juliana, for you and your lab mates for or your lab team, I should say, for doing all this cool work, and for and that speaker series, the beekeeping at home speaker series looks really awesome. And we were just talking about mitigating climate events, and that's a talk coming up. I saw that July 26th, so I definitely plan to go to that. So yeah, thank you, everybody. And now we're gonna give Garrett some time. Dr. Garrett Slater is joining us. Yeah, this is just very exciting to have you with us. And so Garrett will be talking about why are so many honeybee males unable to properly reproduce. And his background is really diverse, which I think will be really awesome to see. How you tied into what you're doing now, because he started 10 years ago working, or he has over 10 years of experience working for a large commercial beekeeper in North Dakota. And prior to taking on a PhD, he also had done a master's on nutrition and queen quality. So this really links right up to it. He also worked with the Bee Informed Partnership for a couple of years, where he worked directly with 30 commercial beekeepers in North Dakota. And his current thesis is focusing on using genomic techniques to understand drone fertility issues in commercial operations. And so he has recently passed his dissertation, and we wish him the best as he now begins his postdoctoral fellowship at the USDA-ARS Bee Breeding Lab in Baton Rouge in October, where he will be applying modern genetic tools to honeybee breeding. So this is such a great complement of a presentation to Juliana's, because we're looking, of course, at like this broader breeding issue, but something that I'm quite fascinated with, and I'll get off my little box here in a minute, is just how that really affects longevity, right? And into the future, these varying traits that can be passed on, or what's upregulated, deregulated. So happy to hear all these talks tonight. All right, Garrett, the floor is yours. Thank you.
Well, perfect. I really appreciate you having me, and I'm really excited to present what I've been doing over the past 4 years. And so, when I first started this PhD, I was really interested in queen quality overall, just, working as a commercial beekeeper and kind of seeing these queen quality issues too with the Bee Informed Partnership. And so, and I really decided to focus on the drone aspect of it, because, we really don't know anything about the drones. If I ask you, like, when you think about like a really quality colony, do you think of drones? It's very rarely anybody raises their hand. And so I decided to really address this question, like, why are there so many drones that are unable to reproduce? So beekeepers lose, such a large proportion of their colonies every single year. I think with the Bee Informed Partnership last year, it was around 45% of their colonies. And one of the major drivers of colony losses is queen failure. Now, a lot of research and energy has really been focused on, the queen aspect of queen failure, right? What is wrong with the queens, and why are they failing at such a high rate? There have been reports that they're failing within, 5 to 6 months, where they should be properly reproducing and highly functional for, 2, 3, up to 4 years. And we're starting to realize that, well, this may not actually be a queen issue. This actually may be a drone issue. We're starting to see that, a lot of these queen failures are starting to be linked to kind of poor sperm and poor drone quality. And so I was really interested in is what is the drone contribution to queen failure? Right? Because if a queen mates with a male that produces high quality sperm, those queens would probably do better long term. And so the key question here is, how much variation is there in these drone fertility traits, and how could this be possibly contributing to the queen failure rates we observe in North America? So what is the source of this variation? If we sampled, 1,000 colonies, and we looked at sperm quality traits, you might have this random distribution or this normal distribution where some males are producing this high quality sperm while other males are producing this low quality sperm. Now, this variation can be, environmental, right? There can be certain beekeeper inputs such as acaricides like amitraz or neonicotinoids that can cause drones to produce low quality sperm. And Juliana's lab has done great work on that part of it, kind of showing that amitraz can cause drones to produce low quality sperm. But there also be like a genetic component to this low versus high quality sperm. There could be certain genes or expression patterns that could actually cause drones to produce low versus high quality sperm. And what's cool about looking at the genetic aspect of this variation is if it's heritable, then there's potential to breed and select for, drones that produce higher quality sperm and higher quality traits. Now, first, I want to explain why I really care about genetics. It's actually kind of funny, when I go to these bee meetings, they always ask me, why do you care about genetics and breeding? Why don't you just focus on management, right? Because with management, we can see these short-term progress, right? We can develop protein supplements and beekeepers can see that progress right away. But for me, I think some of the issues that we face as an industry requires some of this long-term genetics and breeding. It may take 10 years, but I think we can sort of address some of these major issues. And the best way to look at this is just look at these other industries. I mean, if you look at the livestock industry and the crop industry, they've just made tremendous progress in, breeding and selection. And so the question is, how can we integrate this into beekeeping? For example, the average cow, I think, produced, 6,000 pounds of milk 40 years ago, and now it's 24,000. In North Dakota, we could never grow corn in the western part of the state, and now corn is prevalent across the western part of North Dakota. And so I'm really interested in trying to develop these breeding and selection techniques and trying to integrate this into the beekeeping industry. And so one of my major questions is, what are the genetics that's actually causing drones to produce low versus high quality sperm, and how can we actually begin integrating this into the beekeeping industry? And so, if you begin doing that, I think we can start making this tremendous progress that, researchers have been really telling beekeepers for many years. So when you think about a successful drone, we have to think about the entire timeline of a drone reproductive lifespan. And so it spans around 54 days. Some drones can, live longer, but on average, it's around 54 days. And there's three major time points that, from a breeding and selection perspective that we're interested in. Point number one is just sperm production and sperm quality in general. I don't think a lot of beekeepers know this, but sperm is only produced during development for drones. And so once these male drones emerge as adults, they can never produce any more sperm again. And so these early developmental stage is imperative for sperm quality and production. And this usually occurs during the first 24 days of development. And then once these drones emerge as adults, they stay within the brood nest area of the colony, and they receive this care by the nurse bees and the workers. And these nurse bees provide them food, warmth, and all this care. But this point is really important because this is when drones begin to reach their sexual maturity. And so sexual maturity isn't necessarily tied to sperm production, but I'll talk about that a little bit. But these first 10 days, once they emerge as adults, is extremely important for sperm quality and production once they actually begin their mating flights. And then point number three is just mating behavior in general. So once drones reach sexual maturity, they'll begin these mating flights. And so they'll go on these, 30-minute mating flights. They'll go to these multiple congregation areas, and they really do this for the rest of their lifespan until they really either mate or they die trying. And so this whole process takes around, 54 days. But there's three major points here. Thinking about sperm production, 2, sexual maturity, and 3, mating behavior. Okay, so first I'll focus on point number 1. And the biggest takeaway here is that sperm is only produced during development. And this is kind of counter to a lot of taxa or even closely related insects where, like flies, for example, when they emerge as adults, that's when they begin producing sperm, and they can produce sperm throughout their entire lifespan. But for honeybee drones, they produce sperm only once during the larva and pre-pupa stage. And so the gonads begin development when they're in the egg stage, and then they reach full kind of development once drones reaches pupa stage. Now, sperm begins being produced during this late larva instar. So this is when the drone larvae are really fat and big, and this is before the colony actually begins capping the cell. And this is, so the spermatogenesis 1, this is when the first round of sperm is produced. And then once the drones enter this pre-pupa stage, where they're kind of this cocoon spinning stage, the cell becomes capped, this is when sperm is produced again. And so this is the only point in development where sperm is actually produced. And so this is extremely important for drone fertility in general. And then once drones reach this kind of pink-eyed, purple-eyed stage, this is a stage known as spermiogenesis. And this is when the tails of the sperm begin developed. And so the sperm cells are developed during this larva to pre-pupa stage, but the tails and really the quality aspect of sperm is determined during this pupa stage. And so once they emerge as adults, they have all the sperm they need, or they have a fixed number of sperm that they'll use for the rest of their lifespan. And so the really biggest takeaway here is that sperm is only determined during development. And one kind of side note here too is, from a beekeeper's perspective, if this stage is really sensitive for sperm production, what do you think can affect fertility? And this is something I think, probably Juliana is probably addressing some of these questions. We think about amitraz or neonicotinoids or even mites, like these drones are mite magnets. And so if there's any type of negative environmental stimuli acting on this stage, that's probably going to affect drone fertility to some extent. And so I'm focusing more on the genetic aspect of it, but just thinking about this stage in general, I think is really important for a breeder. Okay, and then we get to point number 2. And so like I mentioned, sperm is only produced during development. And so once drones emerge as adults, they have to go through this sexual maturation stage. And so, most of the books say that sexual maturity is reached between, 7 to 10 days. And so what that really means is the sperm, I don't think you guys see this marker? I think you can. But you can see where the testes is pointed at. The drones emerge with all the sperm within their testes. But in order to reach sexual maturity, the sperm actually must migrate from the testes to the seminal vesicles. And so immediately after the drones emerge, the sperm actually begins to swim from the testes to the seminal vesicles. And this whole process takes, 7 to 14 days. And drones don't reach sexual maturity until the sperm really fully migrates to the seminal vesicles. And so once it migrates, then drones reach what we call sexual maturity. And so it's really, sexual maturity for honeybees is more tied to the sperm movement and migration rather than spermatogenesis and sperm production, which is really different than really most organisms, even closely related organisms. And this is what a mature drone looks like. You can see that at the tip of the endophallus, where we have this long endophallus, and the tip, you can see kind of this brown spot, and that's where the sperm is. And so when you pop the drone, you can see this sperm at the tip if they're mature. And this is an example of an immature drone. You can see it's very clear and there's no sperm at the tip of it. And so that's how we sort of quantify, whether a drone was immature or mature. Now, the sperm movement, I think there's 3 major things we need to think about with the sperm movement. First, if the sperm movement is slow, then that could actually delay drones from reaching sexual maturity. And so if it's slower, for example, drones might delay sexual maturity to 14 days. Now, also, let's say sperm movement is impacted, maybe due to chemicals or just due to natural variation. Let's say drones emerge with, 8 million sperm cells in their testes, but only 4 million of these sperm cells are actually able to migrate. Then that means even though drones may be producing a lot of sperm, only half of that sperm may be usable. But also, let's say that they emerge with 8 million sperm cells, but none of that sperm can actually migrate from the testes to seminal vesicles. And so really drones may produce enough sperm, but the sperm may be ineffective at actually moving. And so thus it's not available for mating. And so the sexual maturity time point is extremely important. And I think it's very underlooked. And if you're a breeder, the most frustrating thing is going to a colony and just finding a bunch of immature drones. And so I'm trying to look at that question too. And then point number 3 is this mating behavior. And so, the textbook example of when drones begin their mating behavior is when they actually reach sexual maturity. And so drones will begin their mating flights and they'll go to these multiple congregation of leks. And they usually go to, 1, 2, or 3. And this is occurring in sort of a 30-minute period. And then they'll come back to the colony, go to the outer part of the honey box, refuel with honey, and then fly again. And they'll do this for, 21 to 28 days, really until they either mate or die trying. And I'm sure most of you have seen these congregation areas, but they're really amazing to see, especially when a queen enters them and you see these giant drone comets chasing the queen and really competing for queen access. And she'll mate with 15 to 50 drones during these multiple mating flights. And drones usually find queens based on pheromone or visual cues, but I think there's a lot more that we need to learn about these congregation areas and these mating looks. And so just to summarize, when you think about poor drones in general, we think about, point 1, sperm quality, right? If drones are producing low quality sperm that's, maybe partially dead, then that could affect not only a drone's ability to mate, but affect queen failure and queen quality. 2 is sexual maturity. If drones are delaying sexual maturity, then they may actually have less sperm available, or the sperm may be lower quality in general. And then 3 is a mating behavior. So these drones ultimately had to find the queens and successfully find the queens to mate. And so, one of my major questions is, how much variation is there in these traits, right? How much are the drones producing high versus low quality sperm? 2 is, what is the genetic component to this variation? Are there certain genes? And there's a heritable variation actually explaining this variation? Because if there is, there's tremendous breeding and selection potential. And then 3 is, possibly this is kind of more of a future direction thing, but how can we actually link this? If we find variation and it's heritable, how can we link this to queen failure and queen quality overall? And so we'll be looking at those 3 as I sort of go forward. So first, I'm going to look at point number 1, sperm quality and sperm concentration. So just to summarize once again, sperm is only produced during development for honeybees. And so spermatogenesis 1 and 2 occur during this larval to pre-pupa stage. And then the sperm tail is produced during the spermiogenesis stage in this kind of pink-eyed, purple-eyed stage. Now then, once drones emerge as adults, the sperm is not always available to mating. The sperm actually must migrate from the testes to the seminal vesicles. And the sperm in the seminal vesicles is the sperm that drones can actually use for mating. And so even though drones may produce 8 million sperm cells, for example, during development, it depends on how much of that sperm is actually swimming from the testes to the seminal vesicles. And so for this next aspect, I'm not necessarily looking at how much sperm they produce during development. I'm looking at how much sperm is actually available during mating, because I think that's the most important part when thinking about sperm quality and drone fertility. So in order to answer this question, we did this common garden experiment. And what a common garden experiment is, we have sort of a single environment. And what we try to do is we try to bring distinct genetic stocks from across the country and bring them to a common garden. And we try to control every single aspect of the variation. Me as a geneticist, I'm trying to get rid of as much variation, environmental variation as I can, just so I can understand what are the genetic components to this variation. And so we collected 4 distinct breeding lines from across the United States. And these are commonly used breeding lines for commercial beekeepers. We also did some genetic tests, and they are genetically different. And so we felt like we'd probably see the most variation among these 4 lines. We brought them to Indiana, around Purdue, and we raised them in a single environment. And then we actually raised drones in these 4 colonies. And then once the drones emerged, we put them in one single colony. And the reason we put them in one single colony was because we wanted to make sure that the colony environment was the same, the worker behavior, like the worker care for the drones, was practically the same, and the nutrition was the same. And so we wanted to control for as much environmental variation as we can, just so we can see what is the genetic component to this variation. And then we sampled drones across 3 major time points, 10, 14, and 20. And we did this because the sperm migration kind of followed this kind of reproductive curve, where sperm begins migrating around right when they emerge, around 1 to 2 days, and then it migrates and really maximizes around 14 days. And so you can see right here, this is when drones are producing the highest quality, highest concentrated sperm. And then over time, the sperm begins to decrease due to oxidative stress, and then sperm begins to die. And so what we wanted to do was not only capture the variation from these 4 different lines, but we wanted to capture the variation by age. And so we sampled drones at age 10, 14, and 20. And like I said, again, like they're all from the same colony. And so they're treated the exact same. And then we measured sperm quality traits. And so we looked at sperm concentration. We looked at sperm viability. It's sort of an inaccurate measurement, but when tied to concentration, it can provide a lot of information. And then we looked at other sperm quality metrics, but I'll focus mostly just on sperm concentration because it's highly related to a lot of these traits. And so here are our four distinct lines. We looked at sperm concentration. And so if you look at the y-axis, this is the number of sperm per million. And so for example, this would be 6 million sperm cells. And these are the drone ages 10, 14, and 20. And so I'm putting up the 4 lines in this PowerPoint, but I want you to really focus on line 1 and 4 because these are 2 lines that had the most variation. And these are lines I really want you to focus on as I go further in my presentation. And so for line 1, you can see this, what I like to call a reproductive senescence, where sperm increases over time and then decreases as drones begin age. And so you can see at age 10, line 1 produced around 4 million sperm cells, increase to 6 million, and then decrease to around 3 million. Now for line 4, we see that age 10 was around 3 million sperm cells, increase to 4 million, and then decrease to 3 million. But the biggest takeaway here is just look at this tremendous variation at age 14 for line 1 and line 4. We can see that line 1 produced nearly 6 million sperm cells, but drones from line 4 produced around 4 million. And so there is so line 1 drones produced nearly 2 million more sperm cells, even though they're raised in the exact same environment with exact same conditions, and they were also naturally mated drones. And so what this suggests is that there's tremendous variation, but this variation is likely genetic. And so in order to answer how much of this variation is actually due to genetics versus environment, we looked at heritability. And so the best way to think about heritability is it goes from a scale from 0 to 1. And 1 means that 100% of this trait is determined by genetics. And so a good example of that would be like eye color, where there's certain genes that can cause a human to have blue versus brown eyes. And a 0 heritability would mean that this trait, there's no genetic part of this trait. It's only environmental. And we found that this variation had a 33% heritability. Now, while this does seem low, this is actually really high for a fertility sperm trait like this, where a lot of taxa, like cows, for example, their heritability is around 0.2 or 0.15. And so this is actually really, really heritable. So nearly 33% of this variation was due to heritability. So next we were interested, okay, we see this tremendous variation, especially between line 1 and 4, and there appears to be a genetic component to this variation. Now, the next question is there actually specific genes causing this variation? Because if it's heritable, there has to be certain genes that are actually underpinning this variation. And so we looked at expression in the testes and the brain. And I'm really going to spare you the methods a little bit. But what we did was we sequenced testes and brain tissue from 250 drones. And by doing that, we were able to kind of correlate these expression patterns with traits of interest. And so we looked at expression of the testes and we correlated it to sperm production. And we want to see which genes have higher expression and expression that's associated with sperm production. And so we found a total of 1,200 genes that had an association with sperm production. Now, what's really cool about these, and we like to call them gene networks, there's usually core genes in the middle, and you see this orange gene, for example. And so while this gene network has 12,000 genes, there's usually 1 to 3 genes in the middle that's actually regulating all these genes. And so this is a gene that we're most interested in, because if this gene is sort of regulating this trait, we can use this in sort of a breeding and selection program. And so we found 1,200 genes, we pulled out this gene of interest. Now, what's really cool is we found this gene was associated with dynein. And to try to kind of understand what dynein is, if you're not really familiar with sperm have this head, and the head is where all the nucleus is. And so during the fertilization process, this is when the the DNA is being transferred into the zygote. And then there's this really important mid piece. And this is where all the mitochondria is. So it's essentially the powerhouse of the sperm. But it also has the sperm motor proteins. And so this is essentially the motor to the tail, which is the rudder. And so in order for sperm to move, this motor has to be swimming properly, right? Because without this motor properly working, properly functioning, the tail just won't move. And so that's why this makes this finding really interesting, because it's possible that this could be linked to, low sperm movement in general. And so we pulled up this gene, and our next question was, is there differences in expression between Jones that produce high amounts of sperm versus low amounts of sperm? And so what we found was that Jones that produced high amounts of sperm had significantly higher expression of this dyneid protein than Jones that produced low amounts of sperm. Now this sort of makes sense, right? Because in order for the sperm to actually move from the testes to the seminal vesicles to be used during mating, the sperm must actually swim from the testes to the seminal vesicles. So it's possible that the Jones are producing higher amounts of sperm just have maybe higher motility in general, which we sort of saw with our phenotyping. But it's possible that the Jones are producing low amounts of sperm. There could be something that's blocking that signal, where, like I mentioned, if Jones could actually develop with 8 million sperm cells, but if that sperm isn't fully moving, then they may not have the sperm actually available during mating. And so there could be something blocking that signal where not all the sperm is actually migrating, or if it does migrate, the motility is low in general. Now, trying to link this to queen quality, if you think about this, the Jones are producing low amounts of sperm, like they can still mate and actually transfer this sperm to the queen. But if it has actually low expression of this dyneid protein, the motility is going to be really low. And so it's possible that it may not actually make it to the sperm storage organ, the spermatheca. But if it does actually make it to the sperm storage organ, low motility is linked to, sperm that dies rather quickly. And so the sperm actually may become stored, but it may die more quickly once it becomes stored. And so I think that's a really major question, is looking at, okay, these sperm cells have this low expression of this dyneid protein, but what's happening to it after it gets stored? And so I wish I had that data available, but I think I'll have it in the next 4 months. But we actually looked at that question. But I think that's gonna be kind of an interesting way to link, this drone fertility to queen failure and queen quality in general. Okay, and so just to summarize, we see that especially between line 1 and 2, we see that there's significant variation in sperm number, and it appears to be heritable. And then 2, this appears to be linked to this dyneid protein, right? It's linked to that motor protein that's really causing the sperm to swim and the rate of swimming. Okay, so next we looked at sexual maturity. And so just to summarize once again, Jones emerge as adults, and they're within the middle of the colony for around 10 days, and queens are giving them preferential care. They're making sure that they're at a right temperature, and they're transferring feed. And actually, workers seem to be able to sense drone quality. We know that drones are producing less sperm. The workers will actually give them more preferential care, and so it does seem that the workers to some extent are maybe not regulating drone quality, but trying to improve drone quality by giving them more care before they actually begin their flights. Now, the more important thing here, so on the first section, we see that the dyneid protein expression was really low. And so that means that for some drones, if the expression is really low, one prediction is this sperm movement would actually be really slow. Now, so for the low sperm producers, it's possible that this low sperm motility is causing less sperm because just that low motility in general. And if that's the case, it's possible that drones from this line actually would be less mature, because in order for drones to reach sexual maturity, the sperm must migrate from the testes into the seminal vesicles. And so if the sperm movement is slow or non-existent at all, then drones will delay sexual maturity or maybe never even reach sexual maturity. And so in order to look at that, we looked at immature drones and mature drones. And so you can see right here what a mature drone is. You can see the endophallus is a structure right here, and there's brown at the end of the endophallus with the mucus, and that suggests that the drone is mature. And if for immature drone, you can see this whole endophallus where it's clear and there's no sperm at the tip. And so that's an immature versus a mature drone. And so to look at how much variation there is in maturity, I didn't change the title, but we're looking at how much variation there is in sexual maturity. And so we did the exact same experimental design where we had these 4 distinct breeding lines that beekeepers commonly use. We raised them in a common garden experiment in Indiana. And then once we raised drones within these parent colonies, and then once they emerged as an adult, we put them into 2 colonies to look at the variation in maturity. Then we collected drones at 10, 14, and 20 to capture the variation in this reproductive senescence. And we looked at whether drones were mature or immature at these different ages. And so we marked sexual maturity by this. And so here's a variation between line 1, 2, 3, and 4. And so we looked at what percentage of the drones that we collected were sexually mature or not. And we collected around 75 per time point, because I think we had 3 colonies per line, so we collected around 75. And so this is based on that rate. And so we did look at maturity at 10, 14, and 20. I'm going to focus on 20 here because at age 20, we would expect that drones should have reached sexual maturity, or at least most of the drones should reach sexual maturity at this point, right? Because I mean, if you look at any of these kind of old papers, drones typically reach sexual maturity around 14, or around 7 to 14 days. And so around 20, our expectation was that most of these drones should have reached sexual maturity. But what we found was that line 1, only around 70% of these drones actually reach sexual maturity. But when you look at line 4, these drones that produce the least amount of sperm, only around 30 to 40% of these drones actually reach sexual maturity by 20. Now, it's possible that this is partially due to methods, but these drones were also treated the exact same, right? They're put into a common colony without any type of environmental input, right? And they didn't have mites throughout this whole time, as we're actually looking at that part of it. And so it's really concerning that there's this huge difference between line 1 and 4, where it's like a 40% difference between the percentage of drones that were mature versus immature. And I don't want to speculate too much, but I think this is a really interesting question of why this is. But I mean, it's possible that drones don't reach sexual maturity at this point. It's possible that they actually never may reach sexual maturity, or if they do reach sexual maturity, the question is how much sperm would they actually produce? Because by age 20, this is when the sperm actually begins dying and declining over time. So we see significant variation between line 1 and 4, and our other question was, is there a heritable component of this variation, right? Is there a genetic component of this variation? And there's actually potential for beekeepers to breed and select for this trait. And what we found was that the heritability was a 0.38. And so about 40% of this variation was explained by genetics. And so this suggests to some extent that there's a ton of variation here, but there's a genetic component to this variation. And so kind of back to our question with the sperm quality in general. So we know that there's variation, and we know there's a genetic component variation, a significant genetic component. Are there actually genes causing this variation? And so, like I mentioned earlier, we collected 250 drones, and we looked at expression in the brain and the testes. And just kind of speaking the methods a little bit, we actually didn't find any gene clusters in the testes that were associated with sexual maturity. What we found was that sexual maturity was associated with these expression patterns in the brain. And so, and especially for this cluster, we can see right here, around 825 genes were in this cluster and associated with sexual maturity. And so what this kind of suggests is that sperm production is really tied to the testes, but sexual maturity is tied to maybe these behavioral changes in the brain, where in order for a drone to reach sexual maturity, they have to undergo these transcriptional changes in the brain. And by doing that, they may kind of cause some kind of signal to the testes, not only causing sperm production, but may cause another signal to initiate mating flight in general, because drones really initiate mating flight once they reach sexual maturity. So we found 825 genes expressed in the brains, and these genes seem to be associated with sexual maturity. And so, like I mentioned again, these are part of these gene clusters where the 825 genes associated with this trait, but there's usually a core gene, this orange right here, that's regulating this entire gene network. And this is the gene of interest, and this is one that we're really interested in because these are kind of these high regulatory genes that are really highly involved with this trait of interest. So we pulled out that gene, And the coolest part of this gene it's called RAS32. And you don't have to know exactly what this RAS32 is, but you can think of it as sort of the truck driver. This truck driver is carrying all these nutrients and intracellular material from kind of the brain area all the way down to the dynein protein. And there's a lot of studies showing that this RAS32 is essentially the cargo that carries nutrients to the dynein protein. And so we not only see that sexual maturity is associated with this kind of this truck driver initiative where it moves sperm to the dynein protein, but it seems to be tied towards, in order sexual maturity, it seems to be causing the signal to initiate sperm movement and sperm production overall. And so it seems to be this really interesting pathway between kind of this nutrient production in this kind of cargo container all the way to the sperm and dynein protein. And so it's possible that the reason that this dynein isn't working, this dynein protein has lower expression, is because there's some kind of block in the signal where they're not receiving this cargo from the brain. Now, to actually tie this sexual maturity, we looked at expression of this RAS32 protein between drones that were mature versus immature. And we see that there's significantly higher expression of this RAS32 for drones that were mature versus immature. And so it seems that in order once drones reach sexual maturity, they have this expression of this RAS32. This RAS32 collects nutrients and then it drives it all the way through the body somehow, we're trying to figure that out, to the sperm, and that initiates sperm movement and motility. And so that was a really cool finding, that these core genes were associated not only in different tissues, but they may be causing a connection between sexual maturity and sperm production. And so just to summarize, we see that there's heritable variation in sexual maturity. We see that line 1, such a large proportion of drones were mature at age 20, whereas for line 4, only around 30% were. And it seems to be associated with this RAS32 protein, which is sort of the cargo bus that's carrying nutrients to the dynein protein. Okay, so we see this connection, but we're so seeing that there's sort of some kind of block in the signal. There is expression in the brain associated with sexual maturity, and it's initiating this cargo. But how is that cargo getting to the testes, right? It seems to be carrying nutrients, but how is it actually getting there? And so in order to look at that, we can look at gene connections between the brain and the testes. And so we pulled out these 1,200 genes that were associated with sperm production in the testes. We pulled out the 800 genes that were associated with sexual maturity in the brain. And we were interested, okay, were there specific genes that were involved with both, right? Are there genes that are regulating both the dynein in the testes, right, the sperm motor, and sexual maturity, this RAS32 in the brain? And so we found out that there was around a few hundred genes that were shared between these two gene clusters. And then we pulled out this gene of interest to see what is the connection and is there a block in the signal between sexual maturity and sperm production. And so we saw was that it was this becaudal D. And the best way to think about this is sort of the highway. And so these RAS32, which is just one of these cargo, there's multiple of these cargo that carry nutrients. They're being connected through this protein called becaudal D. And this becaudal D is sort of the highway that's transporting all this cargo and these nutrients and everything the sperm needs all the way down to the testes, that's probably initiating this sperm motor that can cause the sperm to move or not move. And so it's this really interesting path between, reaching sexual maturity where this cargo is being produced, it's being connected to this becaudal D, and it's being transported to the sperm that's initiating sperm movement in general. Now, to actually connect this two traits, we looked at this becaudal D protein, and we looked at whether there's expression differences between drones that were mature versus immature. And so we see that drones that were mature had significantly higher expression of this becaudal D versus immature drone. And so that means that once drones reach maturity, that they're, causing this cargo to be initiated, and that's binding to this kind of this reproductive highway that's carrying nutrients to the testes. And then we try to connect the expression in the gonads, and we see that drones that produce high amounts of sperm versus low amounts of sperm also had higher expression of this. And so, yeah, this is a really interesting link between reaching sexual maturity and sperm production and motility in general. And so it's possible that drones that aren't reaching sexual maturity or sperm production, there could be some kind of block in the system. There could be some kind of link that's preventing drones from not only reaching sexual maturity, but maybe preventing sperm from actually migrating to the seminal vesicles to be used during mating. And so just to summarize, we see that there's a lot of variation, inheritable variation in sperm maturity, and there seems to be a subset of genes that are shared between these. And the gene that seems to be regulating both sperm and maturity could be sort of known as the fertility, this reproductive highway that's connecting sexual maturity and sperm production. So the first two parts, we were focusing on, point number one, sperm quality. What factors are affecting that? Point number two, sexual maturity. And then we try to connect these two traits together. The next part is three. And so one question I had was, these drones aren't reaching sexual maturity until 20, and there's obviously some variation to that. So, but these were naturally flying drones. If they're not reaching sexual maturity by 20, I mean, they must be doing something during flight. I mean, either are they staying in the colony or they're actually initiating these mating flights. Because if these immature drones are initiating mating flights and going to these congregation areas, what's actually happening in these congregation areas? Are they mating with queens, and could that be sort of connected to queen failure to some extent? And so just to kind of summarize, because it's been a while since I kind of covered this part of it, but once drones reach sexual maturity, well, supposedly, I guess, according to textbooks, but, drones begin these mating flights and they'll go to these multiple congregation, sort of, I'll call them licks or DCAs, and they'll fly to 1, 2, or 3 at a time. And they usually last around 30 to 40 minutes, but it really depends on the drone age. They'll come back, refuel, and then fly again. And this will happen for, throughout their lifespan. Okay, so in order to look at mating behavior, once again, we had the exact same experimental design where we had our 4 different lines and our common garden experiment, but this time we collected drones from these 4 lines and we added these small RFID tags on the back of the drones. And then we put an RF reader in the front of the colony. Now, this doesn't actually tell us much. I mean, it tells a little about mating behavior, but we actually don't know where these drones go. But what's really powerful about this RFID tag system is we can track flight behavior. We can know when actually drones begin their flight. We can know how long they're flying, their duration of flight. We have all this really cool data. And this is kind of an example of a single drone I tracked over a 20-day period. And so this is the average length of flight per day. And so for this drone, you can see that for the first 3 days, they weren't flying at all. But then around day 4, around 20 minutes, and then around day 12 or so, they're flying for around 30 minutes. And then, there's a dip here because I think it rained that day. And so this can provide some really, interesting information about just kind of flight behavior. And we could possibly connect this to sort of mating behavior overall. And I wish I had more time to talk about this RFID system. I'm really focused on the sperm part of it, but I could really give a whole 30, 40-minute talk just on the RFID tag system and the flight data I got. But I mean, the question was, all these drones are immature and they're not reaching sexual maturity until age 20. But what's actually happening at this stage? Are they flying? Now, what's interesting is we looked at the age of first flight because our thought process was once drones are, reach sexual maturity, they actually begin mating flights. So at the point they actually begin mating, that's when they are actually initiating mating flights. And there may be some orientation flights, like there's data showing that, but this is really when they begin their flights in general. And we see drones that were mature, they began on average their age of first flight around 6 days. And then for immature drones, it was just a little bit more of that. It was significant, but it was around the 6-day period. And so really most of the drones in our colony, there's some variation, but most of them are initiating their mating flights around at least 10 days. And so even though these drones were not reaching sexual maturity by age 20, they were consistently going on these mating flights. And so we also looked at flight duration, and we see that drones that were mature were flying for around 30 minutes per flight, whereas immature drones were flying around 30, 32 or so. And so they do appear to be flying for longer periods of time. So I'm not exactly sure why that is, but I think the biggest takeaway here is that these immature drones are flying, and I don't know exactly, because it's possible that because they do successfully mate, or they mate with a queen. I don't know what happens after that point, but these immature drones I am collected from the colony do appear to be undergoing these flights and they may be going to the congregation areas and mating with queens. And so, a major question is trying to connect, this immaturity to possible queen quality in general. And so really the biggest takeaway here is that, we see that immature drones do fly, but do they actually mate? That's a question to learn more about. And so I wish I could talk about mating behavior a bit more, because I think that was a really interesting study. But I really think the sperm and the maturity part is probably the most interesting takeaway. And so just kind of summarize, we kind of looked at point 1, 2, and 3. These are these major points as sort of a breeder we would be interested in. First, sperm quality, sexual maturity, and mating behavior. And we see a lot of variation in sperm quality and sexual maturity, and there's a heritable component to this variation. And then we found that there are certain, genes associated with sperm quality and sexual maturity. Now, just kind of take a step back. Beginning the talk, I really focus on the queen quality and queen failure aspect, because when we think about queen quality and queen failure, we can't just ignore the drone aspect of this. We just start thinking about, we can't ignore about that. We have to think about the drone aspect and the mating aspect of it. And so, a major consideration is, how is this poor drone quality contributing to queen failure? And I mean, one simple question is, drones sperm quality changes over time, where it increases and decreases. Queens are mating with younger drones or older drones. What's happening at that point? Or maybe looking more at sperm motility or sperm movement in general, rather than, viability. Because sometimes, sperm can appear dead in these assays, but they could have some sort of motility. And so it's possible, even though queens may be, mating with this low quality sperm, it's possible that it's still actually, fertilizing the egg. So trying to connect that part is going to be a really interesting next point. And next is, performance may be based on location. So I don't want you to take away too much from this, because this is a common garden experiment, and right. So if you think about Indiana, like an Indiana environment, for example, and we've repeated this multiple years and 2 different parts of the season, but it's possible that line 1 was kind of locally adapted to Indiana, and that's why it did really well in our environment. But it's possible that if we move these colonies to a common garden experiment, let's say in Texas A&M, in Juliana's lab, that line 4 may do really well versus line 1. And so this is a question I think, like, me and Melanie have just really been thinking about, is kind of locally adapted stocks, because it's possible that certain genetics may be well suited for one environment, but not the other. And so, this experiment I did last summer, that I'm still analyzing the data, where I spread these colonies across Indiana. And so I'm trying to connect kind of the environmental part of it, because I think it's be a really important takeaway. So I don't want you guys to be too concerned about this until we actually repeat this, multiple environments. But I think there's really powerful methods of using the common garden experiment, but we need to start expanding the environments a bit more. The final thing is, I guess the question is, if there's a genetic components variation and there seems to be line differences here, what's actually causing that? Because I guess one thought process is, if the drones are just producing low quality sperm, or they're not producing sperm at all, I mean, it's not like these genes are being passed to the next generation. And so how can we connect those parts, right? Because if these drones are duds, they're not going to actually, pass sperm to the queen, and that's not going to fertilize an egg. And so one possibility I've been thinking about is these trade-offs between production and fertility. And this is really common in the livestock industry, where if you're breeding for a production trait, or maybe a trait that's not related to fertility, that even though the trait increases over time, we can see right here, for the past 60 years, milk production has increased, but the pregnancy rate has actually decreased. And so a lot of the genes associated with both production and fertility have kind of this inverse relationship. And so trying to find this connection, I think, is going to be really important point. So it's possible that, beekeepers are breeding from one trait, that is having a negative effect on fertility in general. And so this is something I'm trying to think about, why this is. And the final thing is, think about this from a breeder's perspective. I don't know how many of you in the audience are queen breeders or interested in breeding in general, but if you're starting a breeding program, and you're probably gonna be more focused on pest pathogens and diseases. That's kind of like MedHat was, kind of mentioning before I talked, was, should a beekeeper focus on, production, queen quality, or just Varroa in general? And I would say that, Varroa is a major issue. And so that's probably gonna take a priority for most beekeepers. and also measuring for these fertility traits is time consuming, expensive, and just something that's really unfeasible for a beekeeping operation. And so something that I'm really trying to figure out is how can we integrate some of these genetic tools into beekeeping, breeding, and selection. And so instead of using phenotypic selection, where we're looking at the trait, collecting drones and looking at sperm traits, for example, we can sequence a colony and predict which colonies are more likely to have higher versus low sperm production. And so, trying to integrate, we have these genes of interest. How can we be integrating this into sort of a multi-omic breeding program? And so, yeah, if you want to kind of follow my research a bit more, I'll be, I'm hoping to, kind of publish, this paper in the next few months or so. Brock's going on paternity leave, so we'll see how long that's delayed. But I'll definitely share it to your group once it's available. But if you want to, stay in touch with my research, you can follow me on Facebook, you can go to my Twitter or Instagram. And so lastly, I want to thank all the people that supported me financially. And with that, I'll take any questions.
Awesome. Thank you so much, Garrett. This was super interesting. And I see a lot of, just like synergistic questions kind of building off of additional questions, and sort of this never ending rabbit hole we could go down, really finding these connections and learning more about them, between queens and drones and individually too. So that's super interesting. We have a couple questions in the Q&A, and then I saw a couple people had their hands raised. So I'll see, let's get through the question first, because I know I actually also saw a couple in the chat. So let me,
Cool.
I'll go to the Q&A one first, because they've been patiently waiting. And this is kind of just, I'm going to take this as sort of multiple questions, but related more to like just field practicality and the actual production or rearing of queens. So the question is, were the age of the queen, well, it's related to Juliana's in that, sorry, I'm looking at the wrong one. There we go. Here it is for drone saturation. I would like to have 4 different strains of mite-resistant bees involved. Would 4 hives, 1 of each strain, placed at the 4 points of the compass from the breeding yard at about 1.5 miles to 2 miles work? So we didn't really talk about saturation, but I would think that if they're all viable drones, right, there's a shot. But do you have any thoughts on that? They're kind of mentioning here that they'd have a total 16 total drone producing hives. Do you think that's sufficient?
Yeah, it's really tough. I ask this question all the time. It's really tough to answer this because it's really hard to know exactly where the drones are going and where these congregation areas are. Because these queens are flying to these congregation areas, and so it's really location dependent. And so these congregation areas could be 3-4 miles away. And so if you're trying to put these drone producing colonies in one area, it's possible these drones are going to a different area. It's also difficult to know if these drones are actually mating with the queen. And so that's a tough, difficult thing with drones, because we don't know exactly like where they're flying and how many drones we actually saturate the area with. We just have to kind of, try to put as many drones as we can. I would try it. I can't say 100% that it would work, but if you really want this trait to be passed to the next generation, I think trying to saturate the area is the best practice. But if you get 2 distinct lines, I try to put them kind of farther apart, because I think drones typically go to congregation areas within like 2 miles. And so if you're gonna try, maybe do like 4 miles, you might have a better chance. But that's such an open field. I think we don't know much about.
Right. There's so much more we need to learn about that.
It's hard to know where the drones are going to. Yeah.
Right. Hard to know where they're going. And then, well, something came to mind, of course, but now I spaced it. But let me ask this question here. So were the common colonies related or unrelated to the 4 drone lines? Meaning, for example, genetic brothers, and have workers ever been shown to show preference to the brothers, which Liz then kind of piped in about that below with another question. But what are your thoughts on that? So were the common colonies related or unrelated to the 4 drone lines?
No, that's an excellent question. So they were unrelated. Yep. So we had our 4 distinct lines, and the line that the common colony was an unrelated line. And so the thing about preferential care, that's something we didn't look at, but and I was hoping to do this, this experiment where I have kind of observation colonies and actually observe the behavior, because we do know that workers do give preference to care to drones, and they actually get more care to drones that are low versus high quality. And so
Garrett, it's a little bit hard to hear you. I'm not sure if your microphone's getting covered.
Sure. Is this better?
Oh, there we go. Yeah, that's better.
Okay, sorry about that. Yeah, so there's a lot of experiments, especially with I think Michigan State, Zachary Wing did a lot of these studies where these workers actually do give preferential care to drones that produce low versus high quality sperm. And so it's possible that these even though they were genetically unrelated, that the workers in the colony may have been given preferential care to certain drones versus the other ones, but I really can't say that for sure. So I try to control it the best I can, but it's possible.
Right. And then Liz mentioned, yeah, that she typed in her question the same time Etienne had typed in his. But so she says, in your common garden experiment examining drone maturity on day 20 in 3 or 4 different lines of drones, was the one colony they were all introduced to composed of a mixture of workers from each line? And she's asking because we know that lines prioritize queen care for queens they share genetic materials with. So it's possible that drone prioritization of care might happen if the drones from lines 1 through 4 were all introduced to a colony that was, say, line 1.
No, exactly. And that, we really want to control for that part of it, because we would expect that, if the workers and the drones share some sort of genetic relatedness, that they would give them more preferential care. But the colony is totally unrelated. Yeah, but it wasn't even like the same breeding line. It was a totally different breeding line. It was actually the breeding line we have at Purdue, and these 4 were outsourced breeding lines. So,
okay.
Yep. I hope that answers it. Yeah, but it's definitely possible, because I'm guessing if we did do a mixture of workers from these different lines, that they may give preferential care to drones. But I think we would come an interesting next experiment.
Yeah, for sure. That would be really cool to look at. Yeah, that's great. And then I think we had a couple people with their hands raised, and Etienne, are they able to unmute, or should they put their questions in the chat? What do you?
If they're panelists, they can just unmute.
Well, they're attendees, it's telling me here. So
yeah, just Q&A. It's a hassle to unmute people.
Okay, so sorry. So Sandia and Nayan, please, if you could put your comments or questions in the Q&A. I'm sorry about that. I wasn't sure we'd be able to unmute you, but
Garrett,
yeah.
did you test the common colonies genetics and the drones from the common colony?
It says a common colony. No, I didn't. No.
Okay.
Yeah, so you mean the workers in the common colony? What were the genetics of those versus?
Yeah, the workers or the.
No, I didn't, because it is possible they'd be more relatedness to some lines. Yeah. Oh yeah, we never tested genetics of that. Yeah.
A future potential question.
Yeah, well, we did look at the genetics of our 4 lines, and so we did like an admixture analysis and see which proportion of the genetics were A lineage, M lineage, and C lineage. We did that, but we didn't do that for the common colony.
There's always more to do. It's so interesting. It really is. And you're absolutely right. I mean, we need more breeding assessment tools, but also selection tools that are accessible, but also, how do I want to say, sort of user-friendly for producers, And one of the things we were just talking about saturation, it had me thinking of, some of the sort of mecca of queen production in the Western US, which is, in Northern California. And there's so many queens being mated per day. I mean, thousands of queens being mated per day. And you think about how many drones they need to mate with that are, 1, mature, 2, have viable potential. And so it's amazing to think, yeah, bye Juliana, thank you so much. It's amazing to think, why haven't we really focused on that more, sooner? But I'm so glad you're doing this work. This is great.
Yeah, can I mention one more thing?
Oh yeah, definitely, please.
Yeah, and so one more thing too with the kind of the fertility part of it. I mean, it's possible that beekeepers are using all these kind of chemicals, amatraz, especially like during development. It's possible these drones may just respond differently to that. And so maybe selection when you add to the chemicals, right? And so, for example, certain lines, if you add like some amatraz to the drones, for example, they may change their selection indirectly for changes in development, where they may initiate flight earlier, or they may just produce less sperm just as sort of a response, because there's always these trade-offs where drones are just trying to maximize their fertility in general. And so just trying to tie the environment part of it too, I think is be an interesting next point.
Yeah.
Garrett?
Liz, you have your hand up, right? I'm sorry.
Oh, I was thinking the audience might do the Q&A, but while we're waiting for that, Garrett, I loved your talk. It makes me wonder about so many other things, including, so we know that a lot of queens will take orientation flights before they'll take their actual nuptial flights. And so I was wondering if you thought that that may play in to some of these likely non-sexually mature drones flying.
So,
what do you think?
Yeah, so it's tough to distinguish like orientation flights versus mating flights. But one thing we did was we did look at the RFID take system and looked at kind of flight duration over time. And so we see that when drones actually begin their flights around 7 to kind of 14 days, they're around like 10 minutes, and these kind of short, rapid flights. And so to me, that would suggest some like orientation flights. But all the drones I've reached kind of this age 14 time point, like their flights are around 30 minutes. So I would predict that they would have taken the orientation flights already, but most of the drones, even if they were mature versus immature, are now doing the mating flights. But it's really hard to connect these flights to mating behavior, because I don't know exactly what they're doing. But I'm predicting that the ones that are taking these short 10-minute flights, that's gonna be the orientation flight, and then they start doing the longer mating flights.
That makes sense to me.
Yeah.
Ariane and I have some interesting ideas for future studies, so hopefully you'll be.
We're probably working together a little bit, so write it down.
Awesome. Yeah, this is I just feel like there's I'm really excited. Especially because we have such really cool genomic tools coming out, or that are being tested more and becoming more refined, that are really helpful and really allowing us to just better understand what's going on. And what I get really excited about is also looking at it as not just siloing sort of these varying questions, but really seeing where they're interconnected with other aspects, like whether it is due to environment, like you mentioned, right? And then that, of course, is going to link up to nutrition and forage and habitat, which in the broader scheme of things too, also then links up to land stewardship and just the use of the lands and how they're being managed in terms of nutritional aspect for varying, not only pollinators, but people and all the other organisms. So yeah, this is just super exciting. I feel like, we had one more question. You had to mark the drones to keep track of which line they were, right?
Yep.
Yeah. And I'm sure you had to do that also for keeping track of age as well.
Exactly. Well, they're all the same age. I put them in the colony at the same time. But you see, like this drone, for example, you can see like the color. So the screen would be, one line. Well, I had line and colony, I guess. We had 3 colonies per line, and so I had multiple colors. And then the RFID tag, I could distinguish it by then too.
Mm-hmm. Yeah, that's really cool. Just having worked a little bit with RFID when I was doing my master's at Washington State University, it was definitely, folks don't necessarily realize how much time and attention to detail it goes into even just applying these things, and keeping track of that, and incubating these drones and all of that, just to sort of run these experiments. All that minutia of detail is sometimes left out of these presentations, but it's really very tedious. And so it's kudos to you for going through all those steps.
I lost a lot of drones.
Right.
Thanks, Liz.
Yeah, thank you, Liz, too.
I guess we got Ken and Randy. So Ken first, and then Randy, and then that should be good.
All right, great. I had a kind of interesting question because my field, I'm a hobby beekeeper, and I know I don't do it, but I know a lot of people work with their mites by doing freezing drone brood. So is this or could this be? I don't know if you had a chance to look at this or not. Could this affect the maturity of the drones by having more mite load? Or I would have thought so, but
No, exactly. And like I mentioned in the first part, right? Like this early stage is really sensitive to sperm production, right? Because they can never produce sperm again. And like, as you know, like these drones are mite magnets. And so like I'm working with Zaki Lamas a little bit, I'm hoping, because he did some of that work with the mites and the drone side ribs. I like to do more with that, but I'm predicting that the mites have a huge effect, not only the feeding stress, but also the viruses during that stage too. And so, I mean, I try to control it the best I can. Like if, drones emerge with mites, I got rid of them. And, my mite numbers are like had to be 0, because like any type of mite damage could affect my results. So.
kind of a thought. Thanks. Yeah.
It's even possible in the common colony if they had mites, but it's so much I can do. Yeah. Yep, Randy.
I was just curious, that there would be extremely high selective pressure against low fertility drones.
Yep.
So since you're seeing low fertility drones, that would imply that there must be something associated that might be a benefit. Otherwise, that trait would have been completely weeded out whatsoever because of the very high selective pressure. So have you thought about why you would have any kind of alleles that would still in the breeding population that would create low fertility drones?
Yeah, I mean, I think, yeah, because the thing is, like, if they're not fertility, not producing a lot of sperm, like, they're not going to pass it to the next generation, right?
Exactly.
So there'd be strong selective pressure not to have that at all.
Right.
Right. I mean, it's possible that.
So why is it there?
If beekeepers are I think there's 2 hypotheses I have. One, if beekeepers are breeding for a production trait, that there could be an inverse relationship with fertility. So that's possible. 2, actually, for some of these traits, they actually share like genes with queens, for example. So like I'm guessing a lot of these genes are expressed in the testes and the ovaries, and so
that's what I was wondering, whether there's a benefit to the queens and a negative benefit to the drones or something like that.
Exactly. So if you're selecting for queen longevity or queen storage, for example, that could have a negative effect on fertility. So I think that's going to be the next thing we're going to look at, because I guess for twofold, because it's possible they share the same genetics, and so understanding what that connection is. But 2, it's possible you could actually select on the queen part of it, and that could have an effect on the drones. And so we're trying to figure that part of it. But it's possible that some of these genes are inherited from the queen, so it could be just some indirect selection of it. But no, that's a really good point, and that's something that we just try to figure out, because there should be the strong selective pressure against that.
Absolutely.
I'm still trying to figure that out. It's weird. It's weird.
Okay.
Yeah, and it's also possible that maybe in certain environments, right, these drones could do really well, right? Like these drones that produce low quality sperm in Indiana could do really well, like in Texas. So that could be it too.